Hello everyone, I am Dr. S. Karthik Kumar, Associate Professor, Department of Biotechnology, Kamaraj College of Engineering Technology. Happy to welcome you. And now we are going to see the topic industrial innovation. I would like to take you one example how that innovation take place in industries. So here we are going to see how the microfluidic chambers is used as a biosensing element in the field of smart sensor, wearable sensor. Let me explain what is biosensor. It's a device. It's a device as a specific biochemical reaction to detect a chemical compounds in a biological sample. Unless otherwise any biological entity comes into the picture, it cannot be a biosensor. It could be a common sensor, electrical sensor or chemical sensor. So here the biosensor, it has a physical component as well as biological component, where the physical components, it could be a transducer, amplifier, or biological component, it acts as a sensing element as well as it could be a analyte. Look at here, the biological element, it can be either in the form of enzyme, it be sourced from any microbes, plant or animals anywhere, or antibody, nucleic acids in the form of either DNA or RNA, and tissue. Any form of tissue can be used as a analyte here, element here, and microbial. The entire microorganism or a part of microorganism can be used as a element, and polysaccharides. Similarly, the sensor elements, there are different principles involved in sensing the biological elements. Either you can use the electric potential difference in the bioelements, you can use it as a sensor, or electrical current, electrical conductance, electrical impedance, and intensity of the radiations, as well as the difference in the mass, temperature, viscosity. Any principle you could take it as a principle of the test, and you could measure any of the bioelements. This is the pictorial representation how the biosensor it look. It starts with the biochemical conversion, where a chemical biochemical reaction take place, where that the substrate is converted into product, some kind of chemical reactions is come. That time it could be a antigen antibody reaction or enzyme substrate reaction, or the entire cell can respirate that rate of oxygen you could measure, or it can it could be a endothermic reaction or exothermic reactions, where the temperature can be measured, or any aptamers. It could be either the DNA, RNA, how it's hybridized, and due to the hybridization how the molecular mass is get differed. So based on that you could see now it's taking part in the clinical research, aptamer research, and nanoparticles. This is one of the upcoming chapters, the nanoparticles, how it's used in the biosensor part. And the second component is the transducer. Transducer, as we have mentioned, there are different principles can be taken here. Either the electrochemical principle, in which there are different categories. You could choose either amperometric principle, potentiometric, voltammetric, conductometric, impedometric. There are different principles you could choose as the core principle of the test. In the case of optical, either you can go with a color change based, it is called as chromogenic. For example, some reactions it change one color to another color, where the color difference can be taken as the principle of the test. It is called as chromogenic. And illuminants, some reactions it illuminate the light. For example, firefly, you could see that during the night time you could see some kind of light illumination, fluorescent light comes, no. That is a example for luminescence. So that part you can take, or optical fiber. So similarly, gravimetric, piezoelectric, magnetoelastic, there are different principles you could choose as the transducer component. Then very important thing, in the biological system, the signal it's produced by any type of reaction, it will be very meager, it's not able to detect in the larger level. So that time we fail that reactions output. So that's why we need to keep one amplifier, where that signal, the small quantity of the signal, it's multiplied, magnified into quantum, and it's get processed, and the result is get printed in the either in the PC or any form you can get the output. So this is the physical components as well as the biological components, how the biosensor it comprise. Okay, let's go deeper. What is microfluidic? Microfluidic is a small chamber where the capacity of the volume is in the micro level. It is a 1 microliter capacity chamber. It could be a narrow trough, or it could be a small cube or rectangular structures. Anyway, usually the microfluidic chamber thickness it's as like the human hair, 70 micron thickness, that much micro volume it will be processed. The reason for going for the microfluidic chamber, why not that a macrofluidic, you could ask. So, if you look at the property of the liquid flow through a micro channel where that velocity is very higher in the tip of the microfluid. So, while the fluid is flowing, you could see that the velocity is very higher at the tip, and the bottom the velocity is very less. So, it creates more energy, that is why microfluidic chamber is the appropriate one to sensing principle. The another advantage of having microfluidic device is it is very small in size, it is very portable, and at a time multiple samples can be tested. For example, in a small piece of chip you could test more than 100 samples or 1000 samples at a time, as well as it requires a small consensus of reagents and samples. Sometimes we cannot get more samples, for example blood samples, serum samples, cannot get it from the patients. So, only small volume is required, that can also be used in the MFD, otherwise the results will not be convincing. Okay. Let us see the components involved in the MFD. See here, very important thing is the fluid has to move from one place to another place. It is by a mechanical process where the pumps are used to transfer the fluid through the micro channel, where the syringe pump can be used or peristaltic pump can be used. Through the principle of suction and vacuum, the syringe pump it operate, it transfer the fluid through the micro channel. Similarly, peristaltic pump it can be used. The another method is electroosmotic pump, where applying the current you can transfer the fluid from one chamber to another chamber. Very important thing is in the flow of the fluid in a micro channel, the control of the flow is very essential. Usually we will put a valve, ball valve or any different valves. In this micro channel we always prefer the magnetic bead valve as well as the hydrogel valve. Let us see the magnetic bead valve. So, in this picture you would see that the magnetic beads are placed in a pore, and by applying a magnet you can either open or close the valve. So, in which the flow of the fluid can be controlled. So, this is the another type of valve, it is called hydrogel, where that pore is normally closed by applying hydrogel, where the flow will not be there. Once the solvent is allowed to react with the gel, it gets dissolved and it clears up that pores and it allow the fluid to flow. So, this is called hydrogel based valves. So, magnetic valve or hydrogel valve can be used to control the flow of liquid in a micro device. There are different methods to fabricate: photolithography, soft lithography, hot embossing and 3D printing. There are different methods. Let us see few examples. Photolithography is one of the method where the photoresist gel it get polymerized on the surface of the device, and it is allowed to etch to create a structure according to your design. Then it is exposed to the UV, and only the UV exposed area it get stable, the remaining area it get dissolved, it create a structure, it is called photolithographic based device. Nowadays the 3D printing it's play a vital role. So, you can make a design either in the CAD or any software, you can design the structure of microfluidic designs, then you can print it in a 3D design. Either you can go for the resin printing or FMD printing. You supposed to keep a clean room to manufacture the MFD as well as to work in the MFD. So, these are the few facilities is required to prepare a MFD device. And the characterization of MFD it required a multiple test. You could use either FESEM, AFM atomic focal microscopy and TEM fluorescence microscope, UV vis, XRD, XPS. There are different characterization you can go through whether the MFD has been prepared properly or not. Okay, let us wind up the session with one applications. So, here microfluidic chamber for multiple analytes. Let us take the example, a patient is come to the laboratory for testing with the blood samples. He wanted to do a multiple parameters like glucose, sugar, urea, ammonia, different parameters he is supposed to take. So, with a drop of blood samples he test all the parameters in a single chip, that is called multiple analyte. So, where the different reagents are coated in a different chambers in the MFD, a single drop of the blood sample is applied, the blood sample it meet the appropriate reagent, it's get reacted to that reagent and create a color. So, according to the colors you can confirm whether that reagent the analyte is present or not, as well as based on the intensity you are able to see how much quantity of the parameter is there. So, similarly a single chip able to diagnose chloride content, glucose, pH, lactate and temperature. So, this is one commercial application of MFD in the field of healthcare. So, with this we will close the session now. Thank you.